Reproduction in almost all cases in fungi are done by the production of structures known as spores. The spores are the main method of asexual reproduction in case of fungi. The nucleus of these spores contain cytoplasmic organelles. Such as mitochondria, etc. It also contains energy reserves such as glycogen and oils. These energy reserves are mainly here to support respiration during dormancy and germination. In lower fungi, asexual spores are formed in a body known as sporangium. The sporangium is a specialized hyphae in which several mitoses take place to form many haploid nuclei, as we see in this diagram. Sporangium in turn is supported by a hypha known as sporangiophore. The cell wall between sporangium and sporangiophore is known as columella. On the ripening of sporangium, it is seen that the wall thins out. The spores inside the walls take up water, creating outward pressure, thereby making the wall rupture as a result of the great pressure inside, releasing the spores to the outside atmosphere. In ascomycetes, and closely related fungi imperfecti, we see that the production of spores is by a structure known as conidia. Conidiospores may be produced from specialized hyphae which may be singular in nature, or it may be aggregated. These specialized structures are known as conidiophores. Conidiophores give rise to conidiospores. These are either single or aggregated. Aggregated conidiophores are formed to protect the spores from outside harm. In sexual reproduction, the main mode of sexual reproduction is carried out by the formation of gametangia. Here, as we can see in the diagram, firstly the meeting of gametangia takes place, which gives rise to a gametangium. Because of the formation of gametangium, the cell wall between the two gametangia slowly dissolves, which gives rise to cytoplasmic fusion. Cytoplasmic fusion ultimately results in nuclear fusion, but the process is not instantaneous. During cytoplasmic fusion, the cell nucleus are still separated. They are in a phase known as dikaryon phase. In this phase, in spite of the cytoplasm having been fused, the two nuclei of the separate gametangia are still separate from each other, causing this phase where two haploid nuclei are present separately despite the cytoplasm having fusing. But this is just a temporary phase which gives rise to the nuclear fusion, where the two haploid nuclei then ultimately go and form an diploid nuclei. This diploid nuclei undergoes nuclear fusion and thereby causes a mature zygospore. This mature zygospore is formed mainly to protect the spores so that they can be easily used in case of sexual reproduction. The mature zygospore then goes on to germinate. As we see in this diagram, the zygote, which is in a diploid stage, where 2n is represented as a representation of diploid stage, undergoes meiosis to form spores, which are in the haploid stage. Thereby formation of gametangia takes place, cytoplasm fuses, as I said earlier. It undergoes nuclear fusion ultimately through the formation of dikaryon stage, where the nuclei are separated by the cytoplasm is still fused together. But there happens nuclear fusion, we still get the zygote, and as you see, as a result, the cycle continues.